a nice summary of the light reactions visually. And you're seeing that it's in the lumen portion, so it's in the internal portion of the thylakoid membrane inside of it. That there's a bunch of water in there, and that's the source. Photosystem 2 is just really well designed through adaptation and natural selection to take electrons off of water and get them sent into the electron transport chain process all the way to NADP plus. So we tend to say water is the ultimate electron donor. That's what kind of kicks it all off. And then NADP plus is the ultimate electron acceptor. It's sitting at the very end, like, come on, I want my electrons. And then once it gets it, it is converted, reduced to NADPH. And that is one of the big goals. And then the other thing to think about, anytime someone says to you, oh yeah, that's an electron transport chain going in the membrane, then you should start thinking about a proton gradient, because it seems like that's a pretty common strategy amongst all living organisms, that they'll run this electron transport process. And what it does is it provides a little bit of energy at key points to get protons pumped across the membrane. And then don't forget, when you strip electrons off water, one of the products is H plus, a proton. And so between those two ideas there, you get this buildup of protons in the lumen compared to what you see in the stroma. So now it's a proton gradient across the membrane. And that is potential energy. It's available to do something. In this case, they're going to diffuse through the ATP synthase. And as that happens, it actually causes the subunits here to move and rotate, and that causes those two molecules, the ADP and phosphate, to react. And you get that lovely ATP. Proton gradients can be used for other things. It's not just for ATP, but this is a huge goal of all living organisms. Okay. Okay, so we went through that last week. I tried to give you a Google form quiz on that, but we went through it. So now I wanted to get also into more of the Calvin cycle. Are there any questions on that before we shift into the Calvin cycle? So the ATP and the NADPH are being produced in the stroma, which is great because that's right where it's needed for the Calvin cycle. So I believe we left off here where Dr. Calvin and his partners in this research endeavor were trying to figure out what is going on with the Calvin cycle. They knew CO2 is reacting with something, and they know the first stable product is 3 carbons. So they kept looking for that 2 carbon, never found it. So then Dr. Calvin said, what about a 5 carbon? Because then if you bring it up to 6, CO2 and a 5 carbon, now you're up to 6, divide it in 2, now you got 3. That's another way to get 3 carbons. And that's what in fact happened. They were able to discover that it's ribulose 1,5-bisphosphate, RUBP. So that is a 5 carbon sugar. And what happens is it reacts with CO2, and eventually you'll get some lovely sugars. Is glucose the first sugar? No, that's not what the plant's going for. Remember what the plant is going for? What sugar? Yeah, very good. Sucrose. So the Calvin cycle is cooking along in the stroma, and that's right where ATP is getting made, NADPH is getting made. So it's really nice because as the day is going along, the plants are continually making ATP and NADPH through the light reactions, and then the Calvin cycle will take some of those and incorporate it so that the Calvin cycle can run completion. Okay, so it is a cycle. Let's look at it. This is from your book, I believe, and it's giving the visual idea here of the cycle. And the gray balls are carbon, so you can do your carbon counting. So you see here, this is your RUBP, ribulose 1,5-bisphosphate or biphosphate just means you have 2 phosphate groups, okay? And then you see the CO2 coming in to the cycle. And we're going to talk a lot about this enzyme. This is the enzyme that facilitates that first step. It's called Rubisco. And then this is what was tripping them up. Dr. Kelvin and his group, this 6-carbon here, and they say it's short-lived. It's very short-lived. So the technology at the time couldn't pick up on it. It would disappear too quickly in their experimental design. Finally, they got it able to work. And what they realized is that this 6-carbon breaks down into 2 identical molecules of PGA. So PGA, 3-phosphoglycerate, is the first stable molecule of the Calvin cycle. That's why they call it the C3 cycle, because this is the first stable. It's 3 carbons. So C3, 3 carbons. Another way to phrase phosphoglycerate is phosphoglyceric acid. Anytime you see ATE on this, that means it's an acid. So that's where the abbreviation is coming from, phosphoglyceric acid. Okay. Then you can see in that first step where CO2 and RUBP reacting, do you see the artist? Did they draw any ATP or NADPH there? Is it showing up there? No. All right, there's nothing there. So what that's saying is if there's plenty of RUBP and if there's plenty of CO2 around Rubisco, those both go into the active site of the enzyme and react. It's just spontaneous. As long as those 3 players are there, it's going to happen. So that's great. The kickoff to the Calvin cycle doesn't need any push with ATP or NADPH. But then you get into the next phase. Okay, so now you have your PGA going to G3P. That's the next phase. Now you do see ATP playing a role and NADPH. So in order to convert PGA over to G3P, you do definitely need some help from the products from the light reactions. ATP's got to come into play because you need to add more phosphate. You can see the addition of phosphates with ATP's help. And then what NADPH is doing is like, I have some electrons, I can donate those off. And that's what's happening as well. And you end up with the product of that portion called G3P, glyceraldehyde 3-phosphate. Okay, now the last phase of that cycle is being shown as well. So you have G3P going really all the way back to RUBP. You see the way the artist drew it. You see this arrow, arrow, arrow. There's a lot going on there. All right. And they're not showing it. They've abbreviated it. And so basically they're trying to imply there's a lot of reaction steps there. But it's a cycle. So what that means is you have to come back to the starting molecule, which is RUBP. So the Calvin cycle, one of the huge goals is to make sure that it keeps this cycle going round and round so that there's enough RUBP to react with the incoming CO2 during the day. So that's one big goal of the Calvin cycle, just making sure that RUBP is abundant so that the CO2 can react with it. The other, excuse me, the other idea here is in that little green box down there. It says, basically after 3 turns of the cycle, one of those G3Ps that gets made, as this thing is zipping around 3 times, one of the G3Ps that gets made can go off into a different pathway to make ultimately sucrose. All right, so I'll show you that pathway in a nutshell, but that sugar idea is there's a separate pathway they're not showing on this image that will allow for sucrose production. So this is nice to study off of, to do the carbon counting and make sure that, you know, you got a feel for that. And then We use also this image in Plant Fizz because this breaks it down into 3 major steps, which I'd like you to also think about. I think it's a nice logical way to kind of map out what's going on. So this first step, that first bullet point, CO2 and RUBP reacting, get you your PGA. They call that the carboxylation step because you're reacting CO2 with RUBP. So carboxylation, CO2 is carboxylation. So that's the carboxylation step. And that, if that helps you remember what's going on during the first step, I think linking the idea of carboxylation to the CO2 coming in and reacting with RUBP. And then that step is being facilitated by the Rubisco enzyme, which has a big long name. It's ribulose 1,5-bisphosphate carboxylase oxygenase. Thank goodness they put it into an acronym, so it's Rubisco. This is really the most abundant protein on the face of the planet. It's all photosynthetic organisms have this, including cyanobacteria and some other bacteria. Just with plants, it's a huge portion of the protein in the leaves. So that is the enzyme that is capable of running this first reaction with no input of ATP or NADPH. It's just going to work. Okay, then that next step we talked about, going from PGA to G3P, they call that the reduction phase. Right, and they primarily are doing that because NADPH, that's what it does. That's its job. It gives off electrons to reduce one of the chemicals in the reaction pathway. But really, reduction has to do both an ATP step and an NADPH step. And then they actually put on here that, eventually you'll get some sucrose. It'll siphon off. So the G3P is an important, like, branch point, I guess, in the cycle, because once you get enough of that made off, some can be devoted off to let's make some sucrose. And then, but they have to save enough back so that they can build this 5-carbon RUBP. So glyceraldehyde 3-phosphate is only 3 carbons. So you have to go from 3 carbons through many, many steps to get some 5 carbons RUBP. And in order to do this, you will need to add a phosphate group because RUBP has 2 phosphates. Whereas when you start off this regeneration phase, you only have 1 phosphate attached. You got to go from 1 to 2. So you have to push in some ATP. So this last stage, G3P, over to the starting molecule, RUBP, requires ATP. There's many steps. It's called the regeneration phase because you're regenerating RUBP. So between this picture and this one, I think these are good study tools to get your head wrapped around the idea of the cycle and, looking at the carbon, the types of molecules involved. So the Calvin cycle really has 2 big goals. It is, I want to make sure I have enough RUBP all day long so that when my stomata are open all day, hopefully, I can keep taking in CO2, but it has to have some RUBP to react with. And then the other big thing to think about with the Calvin cycle is once they get enough G3P from running the cycle enough times, they can send some off into a different pathway and say, okay, now it's time to make some sucrose. Right, so I'll show you that briefly in a little bit. Yes.
So it's have enough RUBP to keep the cycle going, and then generate enough of the to.
Sure. Absolutely, you got it. So carboxylation, reduction, regeneration. We'll keep that cycle going and going. So as they were studying this pathway, they realized there was some issues with it. And it was very much dependent on environmental conditions, like what is that plant experiencing in terms of temperature, water status in the soil, and they could see that things would kind of fall apart and not be so good. This cycle wouldn't work so well. And so they started studying that a little bit more, going, what's going on here? And they realized that this enzyme that takes runs the first step, even though you don't need to use any ATP or NADPH, it'll just, this will just happen naturally. Rubisco, there's something wrong with it, so to speak. And so what they realized is that we call it a sloppy enzyme because the name of the enzyme has the terms carboxylase and oxygenase as part of the name of Rubisco. The C and the O stand for carboxylase, oxygenase. Remember, anytime you see ASE in a word, that means it's an enzyme. So Rubisco, CO, the C means it can carboxylate. Well, that's what we've been talking about. CO2 comes in, reacts with RUBP, everything's great. But it also has an oxygenase function, which means if Rubisco sees oxygen and oxygen comes into its active site, it's like, well, okay, I'll react oxygen with RUBP. And it doesn't seem to be able to tell the difference. It's like, we call it sloppy because the active site hasn't fully tightened up to just accept CO2 and RUBP. It will take CO2 and oxygen and then the RUBP. So what we realized, or they realized, is that Rubisco can do both. Add in a CO2 to RUBP or an oxygen to RUBP. So if oxygen comes into the active site, that's what you're seeing here on this last big bullet point, RUBP plus oxygen. What ends up happening is you get one molecule of that PGA, which is good because that's part of the Calvin cycle. So that's fine. But instead of getting two identical molecules of PGA, you don't. You just get one, and then you get this funky phosphoglycolate molecule. And this phosphoglycolate is only 2 carbons. Okay, this is only 2 carbons. PGA is 3 carbons, which is what you want for the Calvin cycle. So this phosphoglycolate is a pain. Once that gets made, the Calvin cycle's like, I don't know what to do with that. I don't have the enzymes necessary to work with it. And so the phosphoglycolate gets dumped into a different cycle. That's called the PCO cycle. Another name for it is photorespiration. So some people call the PCO cycle photorespiration. So right now you're respiring. So when you exhale, what is one of the gases that's coming out of your body? CO2. All right, that's why they named it photorespiration, because they see during this PCO cycle, some of the carbon is getting lost in the form of CO2. The plant actually loses CO2 out of its body instead of taking it and fixing, attaching it to RUBP. So this is a problem. You want the plant to take in CO2 and react it with RUBP all day long. You don't want it to give off CO2. That's a loss. Okay. So when they figured this out, they're like, oh, well, this is really bizarre. Why is Rubisco doing that? And basically, it's this idea that here's the blue Rubisco with its active site, and they're showing RUBP coming in, no problem. But if RUBP comes in and then CO2 reacts with it, that's great because you get those 2 PGA and then that feeds right into the C3 cycle. So that's great. But down below here, you see if Rubisco is surrounded by a lot of oxygen, that comes in and reacts and you get 1 PGA, which is good. But then you get that 2 carbon phosphoglycolate, and that's not good. And here's another image showing the carbon counting to prove it to you and the sad faces. All right. So here, if the oxygen comes in and reacts with RUBP, remember now, RUBP is 5 carbons, oxygen doesn't have any carbons, so you're stuck with, you end up with 5 carbons somehow. That 1 PGA is great, but this 2 carbon phosphoglycolate is just not good. The C3 cycle has no way to deal with it. And so now that has to get shunted into a different cycle called the PCO cycle. This is another view of that. If you want an even more detailed view of what's happening with oxygen reacting with RUBP. Okay. So they started to look at this more carefully, like, okay, sometimes Rubisco seems to work really good and take CO2 only. But then sometimes it seems to falter and pay attention to oxygen. And so they started to look and experiment with that, like, okay, how do we figure this out? And so they took the plants and started doing some experiments. And what they realized is that if Rubisco is surrounded by lots of CO2, like if they put it into an enclosure and actually force CO2 into that enclosure, Rubisco would take CO2, run the Calvin cycle, everything's beautiful. But then if they put the plants into an enclosed area and they, pumped it full of oxygen, higher oxygen compared to CO2, then Rubisco's like, well, okay, I'm gonna take oxygen. And then it's like, no, that's not what we want. Calvin cycle starts to falter. You get the PCO cycle kicking in, much less productive. So if you force plants to do this, what's going on the left side here, where you like surrounded by lots of oxygen, keep the CO2 low. And you keep that plant alive through a stretch of time, just doing that all the time, you see that they don't grow very well. Their overall biomass at the end of the experiment is much lower compared to the control plants that are getting what they need. It's pretty much a negative thing for the plant. And so that's why they like, well, Rubisco's ability to take in oxygen, it's not a good thing, because if the plant does that long term, they suffer. They don't grow as much, they don't produce as many flowers, they don't have as many fruits. And so it's considered a not good situation. So let's think about that. So that was in controlled conditions in a lab where they could really manipulate, put them into growth chambers and manipulate everything very finely. But if you're a plant living its life out in the real world, can you think of maybe even around here, there might be times when a plant is experiencing low CO2 in its leaf cells and tissues. So, well, how is the plant getting CO2 in again? Oh, yeah, stomata. All right, so the CO2 is coming in. So what would cause the plant to start to see less CO2? Partly, but let's say it's July, height of the growth season.
It gets really dense and packed with other plants, so it's stealing all the CO2 first.
Partly.
It's really off out when the plant closes stomata. Not taking
Yep. Yeah, that's a big one right there. So especially height of July, right? Drought season, perhaps, hot. Even around here, it can get like that for stretches of time. And so if the plant is like, oh, it's hot today and my roots are sensing I'm in a drought, I have no option. I got to close my stomata, because water, if I dry out, I'm dead. There's a point of no return. So I'd rather close down my stomata, conserve my water, and just hope there's going to be some water pretty quick here. And then I can open up again. And hopefully the plant has enough reserves that it can call on to stay alive for that stretch of time. So in the lab, it was really easy to manipulate that, but under natural conditions, they needed to ask, why would a plant be experiencing low CO2? And it's drought or high heat. And usually it's the combination of the two, drought and high heat, which would cause the stomata to close and conserve water. So then the stomata are closed, there's no more CO2 coming in. So once they deplete the CO2 that's in their cells and tissues, now there's gonna be high oxygen compared to the CO2 levels. And then that's what Rubisco takes in. So what they say is that it's a competitive reaction between CO2 and oxygen. It's competitive. Whatever Rubisco is surrounded by, whatever's more available, that's what comes into the active site. So there's many enzymes that are like that, where it's a competitive thing of what ends up in the active site. And so there's really, Rubisco is probably the most well-studied sloppy enzyme. It's not the only sloppy enzyme out there, though. There are some others, but whatever Rubisco sees more of. I often imagine Rubisco taking a bubble bath, one of those big white clawfoot tubs and all the bubbles. And, it's like, okay, what are the bubbles? Is it more CO2 or is it oxygen? And whatever's more around Rubisco, that's what's going to win and come into the active site. And Rubisco's like, okay, I guess I got to react with oxygen. And that's when you start to get this issue. Does that make sense? Okay. So the PCO cycle. Eek. All right. This is both images here are the PCO cycle. This one's much more fleshed out, where you can carbon count and do all that. This one is much more pared down. I'm not going to get into this, except for a couple key points I want you to understand. Does this look easy to run? No, it's not easy to learn either. So from the plant's perspective, they're like, you're kidding me. I have to kick in the PCO cycle. 3 different organelles. So I'd like you to know which, the 3 organelles. Please know that. Starts in the chloroplast, because that's where the Calvin cycle is. That's where Rubisco is and RUBP. But then some of the products of that have to shift over to the peroxisome, and then some have to get into the mitochondria. And then you see it comes all the way back to the chloroplast. So it starts in the chloroplast, but you have 2 other organelles to think of. And it's a cycle, PCO cycle. So you have to come back around. Then I'd like you to know what kicks it off, which we've been talking about. It's this idea where you have RUBP and oxygen reacting instead of the CO2. And then the 2 products are right being shown here too. With that happening, you get 1 PGA and 1 phosphoglycolate. So that's where the plant's like, this 2-carbon phosphoglycolate, I can't do anything with that in my C3 cycle. I don't have the capability. I have to send it into the PCO cycle. So you can see this reaction pathway, phosphoglycolate goes to glycolate, moves to the peroxisome, shifts to glycine, moves out of there, goes to the mitochondria. And actually this has to run twice. You have to have the PCO cycle essentially run twice to get enough carbons to finally end up with 1 more PGA. Because remember, phosphoglycolate is only two carbons. You have to end up with a PGA out of this whole thing. 1 measly PGA out of this whole cycle. So this has to run twice. And then once you get enough glycine, so essentially what they're showing here, that you see a glycine over here and a glycine here, but they're trying to tell you, you have to run it twice to get 2 glycines down here to do their little reaction. And then you get finally your phosphoglycerate. It shows it over here. They show the 2 glycine coming in. So it's got to run twice in order to go from a 2-carbon phosphoglycolate to 3-carbon PGA. And then once it builds the PGA, then it can stuff that back into the C3 cycle. So that's what we say, it recovers some of the carbon. When we had that previous slide, go back to. Oh, not that one. Where is it? No. Where'd it go? Oh, here. So we say allows for partial recovery of carbon. What that means is you finally get a PGA out of that whole thing that runs twice, involving 3 organelles. And then it says some carbon is lost. So what they mean, I'll show you, let's go back to that lovely image here. What they mean is right here. CO2. Oops, that's not CO2. I don't have my contacts in. See what happened? Come on. CO2, where's the CO2? Oh, man, they should have really highlighted that. Yeah, that should have been like disaster red or something. So anyways, CO2, they say it's lost because CO2 gets cleaved off of glycine in the mitochondria. And then it just gets respired out of the plant, out of the stomata. They say it's lost. It doesn't go back to the Calvin cycle. So it's lost.
Can't it just go back?
I don't know. It's a very good question. I've gone to conferences and talked to plant physiologists about it. I'm like, you sure it can't come back? And they're like,
It's like how it goes into the stomach, the cell next to you, I guess.
Yeah. yeah. And I mean, they can measure it. It's lost out of the plant. I mean, they have like gas analyzers. They can measure it. So it's lost. I know.
Just
Maybe in another million years, it'll figure it out. Plants will figure it out. Rubisco will get better, but so far.
this way for a while.
Uh-huh. It's been, a couple 100 million years. So that begs the question, which is the next slide. Huh, I thought it would do, I gotta fix that, so it's one bullet at a time. So, okay. Plant cells and tissues, you know that the cells are full of water in the cytoplasm, lots of water. If you have water in plant cells and you measure the solubility of CO2 and oxygen in those plant cells, it at room temp, you have to worry about the temperature. So roughly room temp, you have a ratio of 3 CO2 for every 1 oxygen. Okay, so that's how you read out that ratio. There's 3 CO2 for every 1 oxygen. That's good, right? Because that's what the plant wants to work with is CO2. All right, so that's good. And so if you keep the oxygen levels low or they come down, that's great because then you'll have carboxylation, which is good. But if the oxygen levels increase, so it's plant sees more oxygen compared to CO2, then you get oxygenation. So at least the solubility and the amount of gases is tipped towards CO2, but there's like this tipping point where it'll flip and oxygen will get more abundant if the plant closes down its stomata. Then oxygen becomes more abundant around Rubisco. The other issue to keep in mind is that if you increase the temperature of that plant, like on a hot day, whenever you have a body of water with dissolved gases in it and you apply heat energy, it makes the bubbles, it makes the gases come out of solution and bubble out. We've done that test in chemistry or physics. The solubility of gases decreases. That means they bubble out of the solution and now they're not available to the plant. But oxygen is less affected, which means oxygen stays soluble in the water longer with higher temps. So that's again favoring oxygen winning and coming into the active site. CO2 is more easily bubbled out of solution at higher temps. So drought, and now they know that salinity in the soil also can cause too much oxygen coming into Rubisco's active site and kicking in the PCO cycle. So these are these first sets of bullets are kind of just talking about the kind of the chemistry and physics of gases and water, and that increased temp, it can be a problem. So that's why we say temperature is a problem, higher temps around the plant as well as drought. These last 2 bullets here is kind of going back into the big philosophical question of, why did Rubisco even, why is it sloppy? Why didn't it figure out to just take CO2 into its active site and react it with RUBP? But when Rubisco was first evolving, it was actually a couple billion years ago. Before plants were on the planet, it was bacteria doing this. A couple billion years ago. Back then, there wasn't oxygen in our atmosphere. Rubisco never even had to think about it because oxygen wasn't there. And so from that perspective, Rubisco didn't evolve in the presence of oxygen. And some plant physiologists are surmising, thinking, if you look at that complicated PCO cycle, maybe some of these intermediates are useful for other things. Maybe they get shunted off into other pathways that might have a little bit of advantage. And so there's that they're pursuing that idea. So far, I haven't really seen anything that jumps out at me to convince me. And like I said, if you run controlled studies in the lab where you force a plant to do the PCO cycle, they do suffer. Their overall growth rate, biomass at the end of the experiment. So in general, we say this is really not a good thing to run frequently. That's how they think about it. It's not a good idea to run this frequently from the plant's perspective. Okay, let's try a Google form and see if it works. Okay, this one better work. Let me know. It should be a question on the C3 cycle.
Yep.
50-50, my Monday. There should be 2 questions on the C3 cycle idea. You can talk to each other on that. This is fine.
I was thinking about like, because of that, it would all start.
All right. So try to finish up your answer. The second question, you could do it in like 1 sentence. Very good question. And that is something that I have that's why I put that question today, because You need to know the difference between RUBP and Rubisco. And I've seen in the past, students confuse it, and then it's on the exam, that's disastrous. So I'm trying to get that nailed down now. So RUBP is the 5-carbon sugar. If you can remember that Rubisco, the C stands for carboxylase, O stands for oxygenase, O, that's an enzyme. ASE enzyme. So Rubisco is the enzyme. Do not confuse the two. Because we're getting that a lot of points on. Okay, and we'll come back to that. We'll keep going with the C3 cycle.
Sorry.